You know how a tiny cut on your finger just heals itself? Or how a single fertilized egg can grow into a person with trillions of cells? Well, the answer is one of the most fundamental and honestly kind of magical processes in all of life, cell division. And it's happening inside you right now, millions and millions of times over. So let's dive into how it all works.
In this explainer, we're going to break this whole thing down into 5 main acts. We'll start with why cells even need to divide, then we'll look at all the prep work they have to do. After that comes the main event, the incredible dance of mitosis, followed by the final split, and we'll wrap it all up with why this microscopic process is such a huge deal for every living thing.
Okay, so first things first. Before we can get into the how, we really have to understand the why. I mean, why can't cells just keep getting bigger and bigger forever? Well, it turns out, for a cell, being too big is a major problem.
You'd think it would be simple, right? Bigger is better. In the cellular world, that is definitely not the case. See, a cell that gets too large starts to face a crisis of, well, communication and logistics. It just can't keep up with itself.
This right here visualizes the problem perfectly. As a cell gets bigger, its inside, its volume, grows way faster than its outside, its surface area. Think of the cell's surface, its membrane, like all the doors into a giant factory. If the factory floor gets huge but you don't add any more doors, well, you get a massive traffic jam. Nothing can get in or out efficiently.
So it really boils down to 2 huge bottlenecks. First, you get what's called DNA overload. The DNA is the cell's instruction manual, right? If the cell gets too big, that single set of instructions just can't manage the whole operation. And second, just like we saw, is that exchange problem. It becomes impossible to get nutrients in and waste out fast enough. The only solution? Divide.
Now, a cell can't just split in half on a whim. No way. This is a super high-stakes operation. It takes a ton of planning and preparation to make sure both new cells get everything they need to survive. And this long prep period has a name: interphase.
And check this out. This shows you just how much prep is needed. Look at this cell cycle. This huge orange chunk, interphase, is where a cell spends almost its entire life, just growing, doing its job, and getting ready. The actual division part, what we call the M phase, is a really short and dramatic finale to the whole show.
So interphase is broken down into 3 stages. In G1, the cell just grows and does its normal thing. Then comes the most critical step of all, the S phase, which stands for synthesis. This is where the cell makes a perfect, complete copy of its entire DNA blueprint. And finally, in G2, it doubles all its other parts and runs a final systems check, kind of like proofreading the copied DNA, to make sure everything is absolutely perfect before it can move on.
All right, the prep work is done, the DNA is copied, all the parts are duplicated, and the cell has passed its final inspection. So now we get to the main event, the elegant 4-step dance of mitosis.
Now, it's really important to be clear here. Mitosis, specifically, is the division of the cell's nucleus and all the precious cargo inside it, the DNA. The entire goal of this complex choreography is to take that one copied set of DNA and perfectly separate it into 2 identical new sets.
And lucky for all of us, there's a fantastic little trick to remember the 4 stages of this dance. PMAT. PMAT. That stands for prophase, metaphase, anaphase, and telophase. Let's walk through them one by one.
First up, P is for prophase. Think of this as the packing phase. The cell's long, messy, stringy DNA coils up really tight into these dense, X-shaped things we call chromosomes. Packing it up like this just makes it way easier to move around without everything getting tangled. And while that's happening, the container holding the DNA, the nuclear envelope, starts to break down.
Next is M for metaphase. And the hint here is perfect. M is for middle. All of those packed up chromosomes get pulled by special fibers to line up perfectly in a single file line right down the middle, or the equator of the cell. This super orderly lineup is absolutely crucial for what comes next.
And that brings us to A for anaphase. You guessed it. A is for away. Those spindle fibers suddenly shorten, and in this amazing biological tug-of-war, they rip the identical halves of each chromosome apart. One complete set of chromosomes gets pulled to one side of the cell, and the other identical set gets pulled to the opposite side. It's the big moment of separation.
Finally, we have T for telophase. You can basically think of this as prophase in reverse. The chromosomes have reached opposite ends of the cell, and now they start to unwind back into their stringy form. And then 2 brand new nuclear envelopes form, one around each set of chromosomes. So at this point, we have one big cell, but with two distinct, identical nuclei inside.
So mitosis is officially complete. We've successfully made two nuclei out of one, but we're not quite done. We still have one giant cell with a shared cytoplasm. The final act is to physically split the rest of the cell in two.
This process is called cytokinesis, which literally means cell movement. It's the division of the cytoplasm and all the organelles floating around in it. So while mitosis was all about dividing the genetic information, cytokinesis is about dividing up the rest of the cell's machinery.
Now, here's where things get really interesting. Animal cells and plant cells have to solve this problem in two totally different ways. An animal cell has a flexible membrane, so it can just pinch in the middle until it pops into two. But a plant cell has a stiff, rigid cell wall. It can't pinch, so it has to build a brand new wall right down the middle.
So to break that down again, animal cells form what we call a cleavage furrow, where a ring of proteins tightens like a drawstring on a hoodie until the cell splits. Plant cells, on the other hand, build something called a cell plate. Little bubbles full of wall-building material line up in the middle and merge together to form a brand new cell wall that separates the two new cells.
So, we've followed this incredible journey from one cell getting ready to divide all the way to two new cells being born. Now let's zoom out and just appreciate the massive impact of this tiny, microscopic process.
And here it is, the beautiful result of all that work. Two new daughter cells. Each one is a little smaller than the parent cell was, but, and this is the key, each one has a complete and perfect set of genetic instructions. They are, for all intents and purposes, identical clones ready to grow up and eventually divide themselves. So why does any of this matter to you? Well, every single time you grow, you're making new cells through mitosis. When you heal that cut on your finger, your body is using mitosis to replace the damaged cells. And maybe most incredibly, you, all of us, started as a single cell that divided and divided and divided through mitosis to become the people we are today. It is literally the engine of life, growth, and repair. The precision of mitosis is just breathtaking, right? It's a system of nearly perfect copying, repeated trillions of times over a lifetime. But that leaves us with one final really important question to think about. What happens when the checkpoints fail? What happens when this complex, tightly controlled process makes a mistake and stops being so perfect?